It's my pleasure to welcome everyone to the Precision Medicine seminar series. Today we have a guest speaker from UCLA, Dr. Eleazar Eskin. He's a professor of the Computational Medicine, Science, and Human Genetics at UCLA. He's also the founding chair of the Department of Computational Medicine. His research focuses on development of different statistical and computational methods for the analysis of genetic data and advancing genetic discovery for a variety of human conditions. He's also very much involved in educational activities at UCLA. So he's a founding faculty of several academic programs over there, including the Bioinformatics PhD program, the undergrad Bioinformatics minor, the Bruins and Genomics research program, and also the Computational Genomics Summer Institute. He's also a fellow of the International Society of Computational Biology and Alfred P. Sloan Foundation Research Fellow. So we're really excited to have him from the West Coast, and we're really very much looking forward to learning about his latest research.
Okay, so I'm actually gonna talk to you about a really kind of dramatic change in my research area. Last couple of years, kind of really driven by COVID, actually. And so what we decided to do. Okay. What we decided to do is so a group of us basically decided, this is in April of 2020, during the kind of peak of the when everyone got sent home. We also got sent home, and there's this huge testing crisis and all these news articles at the time about how there was no testing. And we were like thinking, this makes no sense, because within our labs, we have all the equipment that we need to do all the testing that we want. And why don't we do something about it? And of course, they let people come back to work if you're working on a COVID project. And so everyone started working on some kind of COVID project. And for us, we were working on testing. And so the question is, why was there such a testing project problem? So part of it was that it's quantitative. The standard technology is quantitative RT-PCR, and this is like PCR test, the gold standard. It's been the gold standard for 20 years, and it works really well. But it's a great technology, and actually, in the end, they ended up doing billions and billions and billions of tests using this technology. But it's not very scalable, so it took actually a lot to scale it. And from our perspective, coming in thinking, what can we do to help? It's also, it's not, when we think of genomics, right? We don't think of quantitative RT-PCR. We think of sequencing, right? So we're curious to say, well, we're kind of coming in kind of from first principles. Well, why don't we develop a sequencing-based diagnostic? And so that's how we ended up with this Swab Seq. So this is basically an idea that we came up with in essentially the end of March of 2020. And the idea was, okay, so if we're going to try to, let's say, do the same thing that a PCR test does, but doing sequencing, how would we do it? So the idea was that, you take a specimen, and the first thing you do is you add barcodes. You do in the PCR for doing the amplification, you add barcodes, and you can mix Okay, do people hear me on the, if I open the chat, I guess maybe I can see, okay. Okay, so now the chat's open, so if you guys wanna let me know, you can.
Yeah, so they can hear you.
Okay, they can hear me, okay, perfect. Okay, so the idea behind molecular barcoding is that for, the PCR reaction is looking for an amplicon, something to that's target. This amplicon is a piece of the spike protein, and the way that the PCR reaction is set up is that when that piece of DNA is being amplified, what's happening is that the reaction kind of adds a little tag of DNA with a specific sequence, and so and we had a different barcode to every individual. So, for example, in this toy example, individual 1 will get 5 A's added to the amplicon. Individual 2 will get 4 A's and a C. Individual 3 will get 4 A's and a G, etc. And so then later, when you mix all the data together and you sequence, you can then look, to see what the barcodes are, and you can separate the data from every individual, and then you can see who's got coronavirus and who doesn't. And the idea for this, the benefit of this is that you can scale this kind of arbitrarily, and we were able to do this allows for sequencing thousands of samples together. So on one instrument, you can do we actually were getting up to over 6,000 samples at a time. And the traditional RT-qC-PCR machine really maxes out at around 384 samples at a time. You've got a PCR requires 2 primers, a forward and reverse primer, that really define what the target is. And in the way that this is set up is that the barcodes are actually hard-coded onto the primer, and they get kind of added to the reaction. So we, for adding different barcodes to different individuals, we have slightly different primers that we're using for every individual, and the difference is in that barcode. So later we're going to talk about how we keep track of which patient gets which barcode, but we'll talk about that in a second. So this is kind of this idea of barcoding. That's one idea. The other idea is this idea of a spike in. So again, the kind of idea that you can use if you're doing sequencing. So what we also do is we basically manufacture kind of a synthetic sequence that looks very much like our target, except for it has 6 bases reversed, which from the sequencer, sequence is really easy to differentiate between what's the real target and what's the spike in. And then we add a known quantity of that spike in to every sample, and then our readout of whether the individual has coronavirus is going to be the ratio between the coronavirus reads that we see versus the spike in. And so the advantage of this is that if there's anything in the sample that'll affect the PCR reaction, it'll affect the PCR reaction for both the true target and the spike in, which will kind of provide, it's like providing an internal control. It's kind of like, looking for allele specific expression, right, versus like total expression, right? So, and so here's actually just showing how well it works. So this is actually a readout. This is an example showing a bunch of samples. So every dot is a sample. On the y-axis is how many reads of the target that we see, and on the x-axis is either contrived samples. So they're kind of how much virus was added. So the 0, those are negatives because no virus was added there. And as you go over to the right, those are progressively more and more virus. You see that, things are working, right? The more virus you add to the sample, the more reads you see of the target. But in general, this is not so accurate because you're going to have to draw a horizontal line. It would be some differentiation between positives and negatives, and you'll see that you'll miss things, right? There'll be some, like if you draw the line here, right? For example, you'll miss, these will be false positives, and all of these ones down here will be false negatives, right? So, however, if we look at the ratio is much, much better, and it's more robust. In fact, if you look at what's down here, there's almost no, these are relatively positive samples, but there's almost no reads there, but the ratio is pretty high. So what's going on here is that there's something in the sample that's preventing it from the PCR from working, but it affects both the spike in. So this results in a very highly accurate test. We developed this based on this. For diagnostics, you have to have kind of a very clear algorithm for deciding what's positive or negative. We kind of make it up actually when you're starting. So this is kind of what we did. If the spike, if you have enough reads, if the ratio is over something, then it's a positive, and if it's a negative, if not, then is there a human sample there? Then it's otherwise, it's negative, and then it's inconclusive. So that's basically the algorithm. So we developed this, and we did this really fast. I think pretty much in April, we had this working. We had some data from the hospital on real samples. We showed that it was working, and then what we did is we went to our leadership in kind of early May of 2020, and we told them, we said, hey, we've solved the testing problem, this big societal problem. Like, look, we have this new assay, we can solve it. Here's our data. And they looked at us, right? Which now, in retrospect, I'm probably thinking, they're looking at us, there's no way. But this is what they actually told us. They said, the genomic technology works, but the logistics are equally challenging. And they said, actually, we're a health system. We actually know how to solve all these logistical problems because that's what we do every day. But there's a pandemic, and so we're really busy. We have a lot going on, so we can't help you. But if you want to just try it on your own without our help, that's fine. You guys can try it. And in fact, they said, there's this. So that's what started this bench to vending machine adventure, right? And in fact, so the first thing we had to do is bring up a high complexity CLIA lab. We need space, money, forms, all this stuff. And so the space is a real issue. And in fact, they told us, they're like, look, they've been trying, it's now going to happen, but they were actually trying to get these billionaires to agree to build this immunology institute at UCLA. And, they were planning on this for a long time. So they had 3 floors of a building that were empty, which would be kind of the starting point for this institute. But of course, billionaires, like they work on their own schedule, right? So it's empty. And so there's an empty floor, and the pandemic was happening with this technology. They said, why don't you just set up your lab there? Because for the couple months that the pandemic will get going, and then you get going. And I think really what they did not want us to do COVID testing in the human genetics building. I think that's the main reason why they were suggesting that we don't set up there. So anyways, we ended up in this beautiful space, sequencers with a view, and we're still there, right? We're still there. The other thing is that we're building the lab, we're buying these sequencers, we're getting started. Then we go to we're trying to get all the supplies. If you remember, the swabs, that was really hard. So we talked to one of the vendors to try to get 2 sample tubes, Thermo Fisher. I said, do you have any sample tubes? They say, yeah, we have sample tubes. How many, oh, that's great. We can sell them to you. I was like, well, how many do you want? I said, well, we were told them that we want 20,000 because we thought that would be like a good amount to start. And then they said, oh, we can't sell you 20,000 because the demand is so high. The minimum order is 800,000. So then we said, how much is that going to be? And so then they said, it's $1.25 million, right? So then we went back to our leadership and we told them this, and we said, like, look, either we buy 800,000 tubes or we buy 0 tubes. Like, this is just really no other options here. And they said, and they said, let us think about it. And then they said, they were thinking like, okay, well, we'll need to do testing for the campus. Why don't we? They said, sure. So next thing you know, like these things are coming in. And so just to give you a sense, this is kind of our summer, spring and summer. We basically, got the assay going, submitted our FDA application, got this, you basically built the lab, all these supplies started coming in, and our first test was deployed in November of that year for UC Santa Barbara. And so what, first of all, you're saying why, but also just I want to zoom in on this. Why is this such a, like, where's the bulk of this? So actually in July, there's this operational pilot with the Ash Center, that's our student health center. So that means that we were actually in July, we were already testing. We were testing, the students were coming in being tested for like coming back to campus. And we were also testing them and showing that the results are concordant. But we didn't have a regulatory license in order to be able to return the results. So that actually 4 months out of the 6 months was really waiting for this regulatory license, right? So we needed to get an FDA EUA. We also needed to get a CLIA license. And this is to give you a sense that, those things we started, in July, and we didn't, and we only got them at the end of October. And so during this time, it was fall starting, UCLA really needed to have its own testing. And with the FDA, you never know if they're going to submit to you in 1 week, 2 weeks, or 3 years, right? So you have no guarantees. So essentially what they did, UCLA ended up actually just signing a contract with another testing. So we're not testing for UCLA. We started testing for other universities. We became a vendor for testing for other universities. And also the willingness of them to how they let us buy the 800,000 tubes really went out the door as soon as we were no longer their campus testing solution. And so we had to come up with really efficient processes to do the testing. And so many ideas, that kind of where sample registration is done at the collection site. Everything is racked and ready for the automation at the collection site. So when the samples get to the lab, they just get kind of put into basically water bath in hot water, essentially, to inactivate them. And then they get put on the automation to make everything very automatic. So this was a really out of necessity because we couldn't hire staff to do the standard kind of accessioning that would be done in the lab. So other things, we're trying to get saliva collection devices because we want to do a saliva test. So they're very expensive and also very limited stock. So actually one of our, Valerie Albaleta, who kind of developed the technology, she actually had this really insight, which is that perfume funnels fit perfectly well into the tubes that we were buying. And we could essentially buy them. So we ended up starting our testing by buying 20,000 perfume funnels on Amazon. And that's really how we got going. Okay, let's skip it. So anyways, just how does this whole how does the whole kind of lab work? So again, this is a new technology, and the idea is that we're doing a diagnostic with sequencing. So we really wanted to think about how we can use the sequencer to help us handle the logistics, right? And the kind of the logistics. So one idea is that all the samples are kind of scanned in and registered at the collection sites. And we use these special kind of tubes that have a barcode on the side that people scan when they're registering their sample, but it's also got this barcode on the bottom. And the racks, they have these kind of holes in them. And so that's actually like all the samples come in like that. And then again, I spoke earlier that, the idea is that we have these primers that end up, these primers that end up, we use a slightly different primer for every sample. So how do we do that? So the idea is that we kind of think of it as like one set of samples. We process 1536 samples at a time, which are 16 racks of 96 tubes. And of course, we could always process more than 1536 or less than 1536, but then conceptually, this is kind of how we think of that as one unit. And so 1536 are those correspond to 4 plates with 384 wells. And so we have these 4 plates, like plate A, B, C, and D. And each one of those, each position in that well has a slightly different primer that encodes a different barcode. And the 4 racks of 96 patients really fit into each plate. So these, out of these 16 racks, 4 fit into a plate. And so you can think of these 16 samples really being 16 racks being together. And you can imagine that every rack has a very specific position, a different quadrant on those 4 plates. So there's really kind of, these 16 racks have 16 different positions on these plates. For example, top right quadrant A is going to be rack 2, right? So, okay. So then how does this actually lead to running the thing? So here's the idea. So we have a sample. So when the racks come into the lab, they get this machine, what's called the vision mate. Actually, it's kind of like a digital camera pointing up. The racks just get put on that machine, and it scans all the barcodes on the bottom and generates a file that looks like a matrix that looks like this, which is in every position. It's what the patient barcode is. And that file is stored. And so, for example, row 3 of column 4 of the barcode will be like this patient sample barcode. And so then again, we have these plates, and we know that, for example, that this position here, row 3 of column 19 of plate C, is this molecular barcode. And then if we know that rack got put into this position where rack 10 is on plate C, row 3, column 7 of rack 10 will overlap with row 3, column 19 of plate C, and the patient sample barcode and the molecular barcode kind of overlap. And we basically store this. So essentially the software kind of takes these files, which are scanned ahead of time, and knows where all the molecular barcodes on the plates, and keeps track of where it gets kind of sent to. And based on that, it generates this kind of database of matching patients to barcodes. So then when we sequence the data, we split everything by barcode. We then run this algorithm by barcode. We get positives and negatives. And then based on that, we can, we go back to that database that we built for connecting barcodes to patient numbers. And that's how, and then we send back the result that somebody's positive. Okay. So that's like the idea behind this. But, people make mistakes. So for example, what would happen if accidentally, like the lab meant to do this, but accidentally they put essentially the liquid from sample 11 into where 12 is supposed to go, and rack 12 to where 11s go. What would happen in this case? Yeah. So what would happen if that happened? So if the lab made an error. Yeah, or like the lab meant to put the samples from rack 11 here, but it accidentally put them into that part of the plate. If they just swap those two by accident. So what would happen?
I mean, essentially, once it's barcode, you'll still be able to identify, right? The position would be wrong, but you'll still be able to.
Close. Yeah. You got a guess?
I'm not sure, but samples wouldn't be as full. They wouldn't be in the right spot.
Yeah, they're not on the right spot, right? So what would happen is that you would think that the samples from rack 11 got the barcodes corresponding to 11, but they actually got the barcodes corresponding to 12. And so what would happen is that the results for rack 11 and 12 would be switched, right? So like if, let's say this one's a positive here, this person would get the result of that positive, right? So it's kind of a big problem, right? Because, well, and then you can imagine that if there's hundreds of racks a day, you can imagine that being not so easy to accidentally miss it. So how do we avoid that problem? So what we do is we leave certain positions empty. If we leave certain positions empty, then what happens is that, we know what positions should be empty, and then we know what barcodes, then corresponding barcodes should be empty. And so then we know that the barcode. So then when we look at the sequencer, we should see that there's no results there, right? For example, right? So later, if we have, for example, these should be empty positions, but then we look in the data file and they're not empty, then we know that something went wrong. So that's basically how we can fix that. And so here's an example of on the wall, an encoding for all like 16 of our racks, and the little orange is like what position should be empty. So you can, so the lab was doing this. They're basically pulling out certain sample positions to make sure they're empty so they could do this. But you can also imagine that this is also not great because you could also make a mistake here. So we actually printed these kind of 3D printed templates that the lab would put over their samples, depending on which rack they are from 1 through 16 to let them know of that. So that's basically, this is the kind of thing that we had to solve, to get this system to work. Okay, so anyways, the supplies all came in, and then we started testing. So we started for Santa Barbara, UCLA Health, Caltech, Pepperdine, Cal State, et cetera. And then we also started. We also got this RADx program found out about us, and we're decided to actually fund us to expand our capacity. And we also started testing over the campus. And so we ended up getting all this like robotics equipment for RADx, which is you know what I'm missing? I'm missing a good slide. Anyways, and so we ended up getting all this robotics equipment, which was supposed to come in, and and then when we got the RADx funding, it looked like it was like the vaccine valley, so COVID was over. But of course, Delta variant came in and back to school, and so we were thinking that but fortunately, like we were going to get like the kind of all the equipment to come in and to do that. So that actually, but how much this talk goes till about 5, right? Yeah. So, but okay. So then, so we're getting all this equipment, and and the campus asked us to start testing everybody when they came back to UCLA, and so that was going to be. So we're like, okay, well, that's great. Well, we're going to get all these robots, and so that'll help us scale up to the lab. But then what happened was that as soon as we signed the contract and paid for the robots, they told us they're only arriving in November, right? Of course, and we're opening in September. So we had to come up with a new strategy to try to test better, like sooner. And so what we it says your screen sharing, yeah. It's not shared on the.
Yeah.
I think they see.
Oh, we can see it.
Yeah?
Yeah.
Okay.
Yeah. Yeah. Okay. 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 So
Thank you.
okay. So anyway, I want to just okay. So we need to find a way to kind of scale up, and so what this is like at the time our test could only do 1536 samples, and so we needed to come up with a way to actually scale up to 6,000, and so or more. So we came up with this idea. So if you remember at the very beginning, I talked about how the primers have 2 components, which is a forward and reverse primer, and they both have a barcode. So the barcodes that we had were called unique dual indexing barcodes. And so the way that they would come to us is that they would come with, the forward primer in large quantities and the reverse primers, and we would actually mix them ourselves to make those plates that I talked about. And so this enabled us 1536 samples to run. And so because we had this crisis, we needed to increase the volume on the plates. And so we came up with this idea called combinatorial indexing, which is that we would mix the forward and the reverse primers instead of like always, A and A, we would make combinations of all the different forward and reverse primers, which would give us up to 16 different combinations. And that gave us, allowed us to get up to 6,000 samples. So then finally, in November, the machines came in and we started having these, this is what the robots that kind of automate our test to allow us to go up to 20,000, 25,000 a day. So this is what it looks like. But of course, when we're installing these, we're thinking that, we'll never really end up using them because, it's not, we really thought that, kind of things were kind of ending at that point. But then of course, Omicron came and, so then that happened, right? So we're now up to about 2 million tests done. So other interesting facts we learned, which is that, for example, we had a for the FDA asked us to test like which of these substances would interfere with COVID testing. And so the first 7 columns here are basically the red shows that there's signal, and this one column has absolutely no signal. We thought it was a mistake. We retested a bunch of times. So there's one of these substances completely blocks any signal. Can anyone guess what it is?
Is this right?
No. Yeah. No, yeah. Toothpaste for some reason completely stops it. Yeah. So if you ever want an inconclusive COVID test for some reason, you get yeah. Okay. So anyways, so what's next? So this was really the first steps of what we were doing. So we wanted to develop, deploy an accurate, scalable PCR test. I think we did that. But then the question is what next? So we're so I think the next steps is this technology. We're using a sequencer, but we're really using a sequencer to really make a better PCR machine, right? That's what we're doing because we're still amplifying a small target and we're just reading it more efficiently than you would using a optical kind of a PCR machine. But really the power of sequencing is not just to do that. The power of sequencing is to sequence in general things that you don't see. So that's what we got actually funded by BARDA and Amazon to develop what's called an agnostic test, which is like a metagenomic test. And so the idea is that it can detect any RNA virus. And so just to give you some perspective, why do people use PCR? So this is the general situation, is that if you're looking at, these samples, like if you get a sample from somebody, like a nasal or saliva sample, 99.5% of all the RNA or DNA is human, and a tiny fraction of it is the virus. And so when you're doing PCR, you're actually targeting it and amplifying it, which makes a lot of sense. So that PCR is a really good application for this kind of thing. And so if you're not going to do PCR and you're not going to target, what are 2 different approaches? One is this depletion approach where you can try to knock out all of the human RNA. Another approach is you can just sequence a lot. So you can just and those are, which one makes sense? It really depends on a lot of issues like RNA quality, library construction, et cetera. And so actually turns out that the sequencing a lot turns out to be better than trying depletion. We tried all these different things. And the reason is because the quality of the RNA is so poor that when you do other things, it affects it. And overall, the sequencers are becoming so inexpensive that you can actually sequence a lot for not that much. Okay, so this is kind of showing more quality. So this kind of led to our newest approach, which is this metagenomic analysis workflow. So you have basically, we do library construction and sequencing, and then you're essentially matching your reads to a database. And then based on that, you're applying recount. So what is it? What is the data? What does the data look like? So here's the so you have 20. You have so this is each row. Here's a sample, and this is one of our previous COVID samples where we know the result, the COVID PCR result. And so you can, and each column here, each entry is the number of reads that we see from, in that category. So actually in this first sample, we have something like 1.425, million reads, something like that. 1.35 are classified in some category. About 1.3 of them million are human, 41,000 are bacteria, and 2,400 are virus. And of those virus ones, 2,100 is SARS-CoV-2. And so based on that, we actually predict that it's actually a positive result. But that just gives you perspective, is that in order to see those 2,000 tests, you have to go through 1.35 kind of million, like you have to 1.4 million just to see those ones, right? But the interesting thing here is that we see the positives pretty concordant. But what's interesting is that, for example, we see that this person is a Swab Seq negative, but clearly looks like they have RSV. So yeah. Well, they're negative for COVID. This is a SARS-CoV-2. This is a sample that we tested for COVID. They were negative, but then when we sequence it, it looks like apparently they have RSV, right? Because there's a lot of reads that have RSV. Does that make sense? Yeah. So I mean, the algorithm works is that it takes every read, it matches it against the database, figures out where that read came from, and so then we see that, for example, for that individual, it looks like 2 million reads came from RSV, right? So clearly this person's got RSV, right? But that's probably why they went and got their COVID test, even though they got a negative result, right? So okay, so then we did this kind of a comparison, comparing to our test, and then what we saw was that we had what we saw is that that it worked, that it worked pretty well. But the interesting thing is that we saw that we found a lot of other things that weren't, COVID. But we didn't have any of the right answers or way to validate this. So we did a collaboration with UCLA Health where they tested for, many different things. And they gave us samples that they tested using this panel that looks for 20 viruses. So they gave us a bunch of different viruses. And so then we can run it and see how we compare against. And so this is kind of our results. So we find most of the viruses that they send us, and these two columns are the results of the device that they're using, which is also not that accurate. So we think we're very comparable to that device, and because they're using this, probably some of the ones that we missed, they might just not be correctly classified. But we also found really interesting things. For example, we saw influenza C, right? So influenza C is something that's considered very mild. And this is actually from a patient in the ICU. So the question is, do we really see influenza C like that? Could that be some kind of an artifact? And so anyways, so I want to kind of take a little segue and say, why is this problem hard? Why, it's hard because, genomic sequences for vastly different organisms could be similar, and also there's incomplete reference databases. So I want to kind of give an example. So let's say, here's a motivating example. So let's say I take my reads and I map them to SARS-CoV-2, right? Just SARS-CoV-2. I might see that 3,000 reads map. Another scenario is I map them to the combined SARS-CoV-2 and human, okay? So what you see is in the first scenario, 3,000 reads map to SARS-CoV-2, and the second one, only 2,000 reads map to SARS-CoV-2, right? So this is something that's really weird, right? Because what happens to those other 1,000 reads? Well, those other 1,000 reads actually map better to human, but in the absence of SARS-CoV-2, they look like they're SARS-CoV-2. In absence of the human reference, they'll kind of map there, right? So if you don't have the right reference sequences, particularly human and all, you'll end up with kind of false positives. So that's the idea is there, you should add more and more references. But there's another issue, which is that you have a kind of like reference contamination. And what I mean by that is, let's say you have some influenza in human and you have a bacteria genome, which, unfinished, meaning unfinished is that they sequenced it and they assembled them and they have these pieces of the genome. They haven't kind of constructed into a full genome. But some of those pieces of the genome are actually not from the bacteria, that they're actually from either a virus or from human, and they're just mixed into the data. And so later, when they finish the genome, they'll try to turn it all into one big circle of genome. They'll identify that some of these pieces don't fit, and they'll correct them and they'll take them out. But in the meantime, in the database, they have some human. And so then human influenza reads might map to these parts, these contigs. And so they're not mapping to the bacteria, but they're mapping to the entry in the genome database corresponding to this bacteria, so that it'll look like this bacteria is present. And so there's like a big cancer microbiome paper that came out in Science that, ran into this summer. They kind of show that there's problems, right? So like, again, if you have too few references, you'll get spurious matches. If you have too many references, you'll start introducing these kinds of problems. So this problem is very hard. So another problem is, if you look at, this is kind of like looking at what the taxonomy looks like. So here, for example, you see that, like in this case, 90% of the samples are human, and there's a huge amount of bacteria, pages and pages and pages of this, because you have all these related strains and matches there. And then even here is a sample that looks very much like they're COVID, but because there's like 250,000 reads that are COVID, but then just by chance, it also maps to some of the other coronaviruses, a smaller amount. So probably this person isn't co-infected with 3 different viruses. It's just probably because of the sequence similarity. So that gives you a question. So in that background of all these problems, did we really see influenza C in that patient? So what we did is we did deep sequencing analysis. We used our larger sequencer for that. And we sequenced longer reads and many more reads per sample. And what we then did was, out of all the reads, we ended up looking, we got 173 different thousand contigs, about 100 million in length, but only 6 of those, are related to influenza C. And, the idea is that, are we confident that it's there? So what we're looking for is some contigs that map, that basically, when they align to the reference, they cover most of the reference genome, and they're very high percentage of what's called like aligned nucleotide identity. The influenza C results. So anyways, so that's what we're looking for. And actually, in this case, there's I think 7 segments of influenza C. We see all of them, and it basically covers that in the lines there. So that's really gives us a lot of confidence that actually what we did see is influenza C. And so here's some summary. So there's 7 matches, and 6 of them are very high aligned nucleotide identity, but it's only 7 out of these 173,000. So we also were thinking that, well, maybe this is in influenza C with some other influenza strain. So what we did is we matched our contigs against all million influenza genomes in the human and we got 1,200 matches. And so this is what the data looks like. So it looks like this is basically the matches, and then there's the data on the strain. So it says influenza C, but they're all from different locations, different pieces of it, because it's the whole million database. That database is kind of a mess. So the question is that, if we have 1,200 of these matches, how can I figure out which organism they all match? Does anyone have any ideas? In 2023, how would you figure out if you have data like this? You have 1,200 matches that looks like this. How would you figure out what organism this is? Any ideas? We can read all of them one by one and see what it is, right? But like a kind of more automated way to do it. No ideas? Anyone on chat? On Zoom? On Zoom? Any ideas? Okay, so it's this command, I'll give you a hint. Do you know what this command does? Yeah. Yeah. So what I did is I took all the text, right? I put it into ChatGPT and I asked, what is the organism? It told me it's influenza C, right? So yeah, that worked. Okay, so anyways, next, we're planning on, we've done respiratory and saliva, and now we're going to go to, blood, other organisms. And actually, I actually have 10 minutes. Okay, so I want to talk a little bit about like what are the kind of computational problems that are here as well, just to give you a perspective. So, the general computational problem is that you have a bunch of reads, and then you have these genome sequences, and you have, basically millions of these genome sequences, and you want to find which read is from the genome sequence. And in general, the way that people do this is that they break up the reads into k-mers, okay? And then for each k-mer, they generate a database which has basically the k-mers, and it tells you which genome sequence has that k-mer. And then using some, however it matches, they somehow add up things, okay? And then based on that information, they make a prediction that this read came from that genome sequence, right? So that's it's really all these algorithms do. They're just a giant index. The difficulty is that there can be petabytes of sequence, right? That you're trying to index, and there's a lot of k-mers, and there's many genomes. So this is actually not so easy. But there's been a huge revolution in the field that make things really almost all the new algorithms for these genomic problems have become very fast and efficient. And it's really because of this technique called minimizers. Has anyone here heard of minimizers? So I'm going to spend a few minutes talking about that because they're really cool. So basically, what's the issue with minimizers? So minimizers make everything fast, and the intuition is that overlapping k-mers are redundant, right? So if you think of these k-mers, you shift the k-mer over by one, you don't get a lot of additional information, right? And shared k-mers also have similar sequence, right? So let me kind of explain what is a minimizer. So let's say you have a 31 mer, like a k-mer of length 31. The 7 mers of that 31 mer would be just if you take a sliding window and you take all of the k-mers of all the sequence of like 7 from there. And then if you sort it, the minimizer would be the first one alphabetically. So if you take all those n-mers, the 7 mers, you'll have a set of like 20, 26 of them, something like that, and you sort them. You take the top one, so the minimizer will be just this sequence. This is so the idea is that the kind of the real original application of k-mers was minimizers was when people were trying to do. Actually even before that, but let's say you have a length of a read of length 100, and you're taking all of the k-mers of length 31. So you're going to have about almost 70 k-mers of length 31 from that read of like 100, right? Because you start at the first position, 1 to 131, then 2 to 32, etc., right? And so you'll end up with about 70 of those, right? But if you look at the minimizers, you have a much smaller number of minimizers. Why? Because if you have two sequences, one k-mer, like, and the next k-mer, they very often will share the same minimizer, right? So if you imagine you're going across the some sequence, when will the minimizer change? There's two scenarios, right? Either you'll actually leave the window of the minimizer and then you'll end up with a new one, or you'll basically introduce a sequence that's alphabetically ahead of your current minimizer. Okay, so the real original application for minimizers was people wanted to count k-mers, and they wanted to do that very efficiently, and they wanted to store the index on disk. And so when you're, like using this technique, they would load, for example, the like they assigned an index based on the minimizer. So they load that index into memory, and they would kind of slide along, and they would get multiple k-mers that had the same minimizer. So then they didn't have to switch the index, because every time you switch the index, you have to save it to disk and then load the next one. So that was the original idea of minimizers. And so if I take this, if I take this index for minimizers, right? The idea is that don't store all your k-mers, just store the minimizers and where those minimizers occur. So the index would look just something like this, right? So many fewer entries, because you're storing fewer, and when you have a read, you just compute your minimizers. You have many fewer minimizers to look up, so it's much more efficient. So the question is, when does this work? So it'll work if at least one k-mer, right, must not have a change. So that way, like if you have mutations and errors, you'll still be able to match what you previously didn't match. And so I actually used ChatGPT to build me a simulator of minimizers to test this out. And just to give you a perspective, for example, if you have like a million length string, you're looking for k-mers of length 15, but minimizers of length 10. Only 30% of the genome will be minimizers. So you can imagine that if you're storing this in memory, it'll be a third. And if you look at different values of K and M, there's like this is actually a good one. K-mers like 21 with M of 15. You're saving about 1/4 of the space by storing minimizers. And for that same one, even with a small number of errors, you're only losing like a very small percentage. So anyways, just I kind of don't want to spend too much time on that, but just give some perspective that that there's a lot of kind of new interesting work there. Anyways, so this is the lab that did a lot of the experiments, and this is kind of the core team of the project. Okay, thank you.
Any questions? So I think we all had some traumatic experiences setting up various, operations during the COVID pandemic. What do you think is kind of the key lesson in terms of setting up a sequencing operation like this?
Well, so we actually just recently had like a little thing for our 3-year anniversary. And we had like the guy who was in charge of making all this decision on the campus, the one that greenlit us for the 800,000. So he said, he's like, they seemed like they could do it and they really believe that they can do it. He's like, but frankly, we were completely desperate. So that's really why they greenlit us, right, for this thing. So I think that's, I think it's, there's a lot, but I mean, I think that's kind of why it worked out. But I think also, if you have, I think this is, you see this in many different contexts, is that if you have a group that's very focused on one thing and they're very much aligned, and get along, they can do a lot, right? As soon as you don't have that, like nothing, very little happens, right? So I think that's like, this is a great example. Our core group, we had like this one focus, and we got along well, and so we went really fast. And so that's kind of I think that's the other, but we all know that. I think that we just, we don't realize how. I guess you don't really think that it's as makes as big of an impact as it does. And so, if you take that to the limit, you should really kind of put all your energy where you're aligned with people and very little into where you're not.
Any quick mention, you mentioned all the potential new sampling that you can do in the future. I may have missed it, we did have fecal stool samples out there, correct?
Yeah, we have, we can, yeah.
Yeah, but so that's something that you will plan on doing, or?
Yeah, well, we got funded, we got some funding to extend to other samples, right? And so, I think that, I think that, we got funding to extend to other samples, and so we have to pick some. That one is just there's a lot already there. So we're trying to pick sample types where there's less, like blood will do because we have lots of blood. But just where there's more kind of demand, right, and opportunity, right? Because people do metagenomics for stool samples all the time, right? There's, so that's something that's not that new. But maybe we will, yeah. Okay. Oh, I see. So, what's the benefit? Okay, good question. What's the benefit does contig assembly offer compared to using sequence alignment tools? Yeah, so okay. So I actually think of it that there's really two different paradigms for like metagenomics. One is you take your reads and you classify them into, or you basically align them, you find them like where they came from. And then the other one is you do meta assembly, and then you take those assembled contigs and then you align those contigs against the reference, right? So they're like, okay, so obviously if you have a huge amount of data, you can generate grid assembly, like the assembly one is better because there's more information, right? Because in somewhere you have more of a sequence, like to help you with ambiguities, right? So, but then, it's also like, they're very complex methods. So there's, you don't really understand exactly what's happening, right? But I think people really use both, right? For that reason, because there hasn't really been a good, like they kind of give you different things for things that are very like, not like very like small number of reads in the sample, then, the kind of classification or alignment of the individual reads is the only thing that's going to work, because you're not going to be able to generate an assembly, right? So we think of the assembly as mostly for helping us explain surprising results, like the influenza C, right? We didn't believe that it was there. I mean, it's just like, like I looked it up on Wikipedia, right? And according to Wikipedia, it's either not infectious or very mild infection, right? So, is this real? Like, there's so many reasons why this could be a false positive. So by getting the whole genome assembled, without it looking at any other data, and then noticing that turns out to be very close to influenza C genomes, that was giving a lot of confidence that it's actually influenza C, right? But, we were able to predict it without it, right? So.
Are you still testing for COVID now?
Yeah, we actually still are. Yeah. We got funding from the state to do testing.
I don't see any other questions. So thank you so much. That's after 5 o'clock.